How many of you are going through the church reading plan this year? Okay. Well, I was really struck by one verse here in 1 Thessalonians. It's chapter 3, verse 13. It says, "May he strengthen your hearts so that you will be blameless and holy in the presence of our God and Father when our Lord Jesus comes with all his holy ones." And I just had this image in my mind of this glorious return of the Lord Jesus when he comes again. And so that just, I don't know, that kind of grabbed me. And of course, 1st and 2nd Thessalonians, the return of Christ, is one of the big themes in that. So here's a picture of Earth and Moon taken from 4 million miles away by a spacecraft on its way to Jupiter. And what I think is interesting is the contrast between the Moon and Earth. So current theories of the formation of the solar system is that the Earth and Moon were formed at the same time out of the same stuff. They bear the same fingerprint, for the fingerprint they use for detecting whether something came from Mars or from certain groups of asteroids and things like that. They have the same fingerprint. And yet, look at the contrast between them. One world totally dead and the other just so bright. And the difference, of course, is life. Life happened on one, it didn't happen on the other. And one of the surprising things as we have gone out into the solar system and been looking, well out into the galaxy and beyond is not that we found life, but that we haven't. And that's really been unexpected. And so there's something very special about life. So you take a living being, and we do this all the time, called cremation, right? When somebody dies, you cremate their body, it all turns to ashes and to the basic elements and stuff, and those elements are the same elements that you find all over the universe. And yet somehow we can do that, but we can't go the other direction. We can't take all those elements and form them together into living beings. And so there's something very different about life. So take this rock, right? All the elements that we're made of is contained in this rock. And yet this rock is inanimate, and we can distinguish very easily between this rock and what life is. So I want to discuss during this class kind of the wonder of life and how life is like nothing else in the universe shows the hand of God in its design. So life, one of the differences between life and rocks is life is purpose-driven, right? These plants sending out roots to get moisture, they're sending up stalks to reach the sunlight, they're producing flowers for fertilization, and they're producing seeds, and then to continue on. So they're kind of purpose-driven, and that's true of all life. It's not inanimate, it's striving, or it's moving along with a purpose. And then life responds and adapts. So this rock doesn't really respond to anything unless, of course, I apply the laws of Newton's mechanics to it, then it moves in accordance with those, but it's really not responding and adapting to its environment in the sense that life does. So you see that little microscopic creature in the lower right, it's called a euglena, and it has so when it swims, and you'll find it in ponds, and it has a little propeller called a flagellum, and it swims around, but when it's too narrow for it to swim, it can start crawling. And that's what you see here. It's crawling between two plates of glass, and then it's swimming, and then the channel gets narrow, and it starts crawling again. So life has this ability to respond and adapt to its environment. And finally, life has the ability to reproduce. And rocks don't reproduce, they crumble down, but life reproduces. And when you look at what it requires to do all these things, and especially with the reproduction, it requires unfathomable complexity, elegance, and sophistication. And this is what life is. So we're going to be exploring this a little bit. So I want to give you a little biology lesson. Okay? 3 slides. That's it. Okay, number 1 is that the fundamental unit of organic life is the cell. All life is made of cells, and there's 2 basic kind of cells. There are prokaryotic cells, so that's what bacteria are, and that's characterized by a membrane with DNA inside of it. So, and the DNA has all the information, and the DNA forms loops. But there's no other membranes inside of it or compartments and stuff like that. So that's bacteria. That bacteria is a prokaryote. And then you have the eukaryotes. And those are the cells that we're used to. They have a membrane, have a nucleus. They have all these organelles, which are little compartments that do different functions. So eukaryotes are really quite sophisticated compared to, say, the bacterial cell. And eukaryotes include animals, which are defined by squishiness, right? That's one way, it's squishiness, right? And that eat things, by the way, whether plants or other animals. And then there's plants, which are characterized by a cell wall, and they can utilize energy from light. And so they're more rigid, they're not squishy, right? And then you have fungi. I'm always pronounce things wrong. But anyway, you have fungi. These are also eukaryotes, but they have a cell membrane, but they also have a cell wall, but a different kind of material than the cell wall that's in plant cells. And they're the decomposers. So basically, they eat other life that's dead, and they eat it by digesting it on the outside, and then they suck in the nutrients. Whereas animals ingest the thing and break it down inside. And so anyway, that's a brief description of the basic unit of life. Well, what about viruses? Viruses really aren't life. They're information packets. So what you have is you have a bunch of RNA or DNA, which encodes information. Right? And then it's surrounded by a protein shell. And so it's not living in the sense that it doesn't have metabolism, but it reproduces not by dividing and conquering like other life, but forcing life to reproduce it. So those little spikes that you see on the outside of the protein shell, when they contact a cell wall, the right cell wall, then the cell wall they recognize each other, and the cell sucks it in. And then the protein shell opens up, releases its RNA, and now the machinery inside the cell duplicates the virus. And it keeps duplicating until the cell bursts. And so that's what viruses are. We hate viruses, right? They're also very beneficial. They have good viruses and bad viruses. Sometimes the viruses that are good for us are bad for bacteria. We'll just talk about that next week. Okay, so that's about viruses. Okay, well, what about the building blocks of life? We talk about the basic elements. Well, you've probably heard of amino acids, fatty acids, carbohydrates, nucleobases. Except for the nucleobases, you've heard of all these in terms of dietary. Well, this has so many amino acids. I got this because it's rich in amino acids. I got this because it's rich in carbohydrates. So what your body does is you eat your food, and then the digestive system breaks it down into these elemental molecules, shall we say, and then those molecules are absorbed from your digestive tract into your bloodstream, and then they flow in your bloodstream to the cells in the body, and the cells reassemble them into proteins, lipids, polysaccharides, and DNA and RNA. So that's kind of the way it works. And John, you can correct me afterwards. Okay. All right, so I want to give you a sense of how small life is, because life really is a thing of the very small, the opposite of astronomy, right? We use telescopes, now we're using microscopes. All right, so looking at the glory of God in the very small. So to bring the elements of life up to a place where you can actually see them, I've taken this guy Max and I've maximized him to a scale factor of 60,000. So I've increased his height and width by a factor of 60,000. All the molecules and elements and atoms have been increased by a factor of 60,000. All right. So now he's looking down on Cleveland. Okay. So you can see Burke Lakefront Airport up there, and you can see its runway. That runway is I think around 1.2 miles long. Okay. So here's his hand. Actually, it's not his hand, it's my son's hand, so I had to upscale, I had to maximize my son Joseph because I couldn't take a picture of Max's hand. So anyway, you can see the little finger is a half mile wide. So watch out, and don't look out the window, okay? All right. What about Max's hair? If you could find a hair on him, okay, he's bald, but if you found a hair, it would be 14 feet in diameter. All right, what about cells? So one of the largest cells in our body is skin cells. How big is one of Max's skin cells? Well, it's about 6 feet wide. So that's equivalent to my height, okay? So he's a eukaryote. So, and I don't know if he does karaoke, but he is a eukaryote. But anyway, so here's a cell nucleus inside a skin cell. It'll be basically 2 feet in diameter. So take one of those exercise balls that you like to sit on and bounce around. It's like that diameter, so that's 2 feet. So that's the nucleus of the cell. Okay, now we're going to get smaller than that. By the way, I want to point out Max, so you can see his foot. Mount Everest is just a little bit above his ankle, and so is the typical altitude for airplanes that are flying. And he could stick his foot in the Marianas Trench, no sweat at all. So his trousers, his lower part is protected from meteors, but not his torso. He's at risk for impact from meteors. And he has no air because his nose is above the edge of space. So anyway. All right, what about his DNA? So DNA is the information-bearing molecule inside of the cell, and so we have our DNA is packaged into chromosomes, and those chromosomes are made of individual strands of DNA. So if you take an individual strand of DNA, it would be the width of a piece of paper. So think of a very thin piece of thread, and it would stretch, that single strand would stretch from Grace Church all the way beyond 130th Street down Pearl Road, that little tiny thread. And that's packaged inside of the exercise ball, okay? Okay, now what about all the DNA of cells? Because he has, 46 chromosomes. So if you take all that DNA and stretch it out, it would stretch from here to the east gate on the Ohio Turnpike. What about bacteria? So we talked about bacteria. So because the prokaryotes don't have a cell nucleus and all these organelles inside, they can be smaller, and so a bacterium E. coli, which hopefully you don't have in your tummy, but maybe you do. Maybe you have some safe kind, inert, benign kind in your tummy, but that would be 2.5 to 5 inches long, would be a bacteria. So compare that to the 6-foot cell, right? So 2.5 to 5 inches long. Apparently, most of our cells in our body are not human, or at least half of them aren't. So they're bacteria and viruses and fungi, and by and large, they're beneficial for us. If we didn't have them, like when you take antibiotics, kills them off, then you have a lot of digestive problems, and you can have other problems too. So don't wash your hands too much. Okay? All right. What about a virus? Well, a virus is like 1/16 or 3/16 inch. So think of a 1/16 inch drill. That's the size of a virus. Okay. So very small. Okay, but we're going smaller still. What about the cell membrane? So the cell membrane is made of lipids. There are 2 layers of lipids that oppose each other, and I think they're hydrophobic in is something. Anyway, I want to explain it. I think Linda, who has a lot of training in biology.
Once did.
Well, you cook, right? Okay, so so you have design expertise also. All right, so the thickness of a cell wall is on the order of a 64th of an inch. Okay, going still smaller, we talked about the width of a strand of DNA. So here you have an electron microscope view of a loop of DNA, thickness of a thin sheet of paper. Okay, now amino acids. Amino acids are what your proteins are made of. Your proteins are made of chains of these amino acids. And we call it residue because it's an acid, I think, when you dissolve it in water. When it's not in water, it's not an acid anymore. I think that's correct. But anyway, again, afterwards, Al can correct me if I'm wrong. But anyway, it's about the thickness of a sheet of regular aluminum foil. Yeah, Ginny?
So it's only that thick if you were as big as...
If you were as big as... Actually, ours isn't. Max's DNA is his. Yeah, that's right. So that's how big his amino acids are. Ours are a factor of 60,000 smaller than that. So anyway, I thought that would be fun, and so now we're going to show this video that I made several years ago. It kind of forms the backbone of this class, so I will close this up, and hopefully this will work. This is part 3 of How Science Affirms the Bible, and so today we're talking about biology. So the issue we're talking about today is the impression of design in the natural world that we see, in particular life. Is it an illusion or real? Because design is something that we naturally perceive. There's an article in Wall Street Journal several years ago by a psychologist, and she said, by elementary school age, children start to invoke an ultimate godlike designer to explain complexity of the world around them. Even children brought up by atheists. Now, I think this gal herself is not necessarily a believer in design, because her solution was to come up with these picture books to explain Darwin. Of course, Darwin is the one who came up with the materialist explanation of life. And if materialism is true, then there really is no design. It's all random, it's blind. So really design is an illusion rather than real. And in fact, Darwin explained that as he expounded his theory on origin of the species. Richard Dawkins himself has said that's what evolution does. Darwinism is it explains how there could be the appearance of design when in fact there is no design. And well, what's the issue? Well, design, inherent in the meaning of the word design, it means there's a designer. And a designer ultimately means God. And where there's design, there's also purpose, or teleology is the fancy word for it. And so that's kind of what the issue is. So I have this neat quote I got. It's from this book called Nature: How Mind Emerged from Matter. So again, it's kind of another materialist type book, but it says teleology, or purpose, is like a mistress to the biologist. He can't live without her, but he doesn't want to be seen with her in public. So anyway, so let's give a little background on the history of biology. Is design really an illusion, or is it real? Is it really there? So biology is a thing ultimately of the small, the very small, whereas astronomy is the opposite, it's the very large. So see the invention of the microscope in 1590, but useful microscopes really weren't available until the mid-1600s, when microorganisms were first discovered. And then it was in 1839 that the cell theory of life was proposed. That is, all life consists of cells. Cells are the fundamental unit of life. You might say the atoms of life. So it was 1839. Well, in 1859, Darwin published his Origin of the Species, the theory of evolution, as we call it these days. But he didn't come up with this theory out of the blue. A number of people have been thinking through that. Another guy by the name of I think his name was Owens. I'm sorry, my name is blank. But also proposed a theory at the same time. In fact, Darwin raced to get his publication. His theory published before this other gentleman could do it. But they, as the age of exploration, they explored the world. They could see how species varied from place to place, and paleontology was growing. They could see how species varied from time throughout time, and so forth. And so it's not like he didn't come up with this out of the blue. But the significance of his theory is that it came up with a naturalistic or materialistic explanation for life. So it came up with an explanation for life without design, without intent, unless you would say that the intent was in the design of the original laws and constitution of the universe at its beginning. And so that's the significance of his theory, is that it came up with a materialist explanation of life. What's interesting is what happened around the same time. So it was in 1860 that a guy by the name of Clausius proposed the law of entropy, which does present problems with the origin of life. And then we had Louis Pasteur in 1862. He experimentally disproved the theory of spontaneous generation, that is, that organisms, microorganisms in particular, could form spontaneously out of garbage and rotting vegetables and stuff like that. So he proved that was false. And then shortly after that, an Augustinian friar by the name of Mendel developed and published his laws of inheritance. Those remained obscure for some time, basically misunderstood and ignored until the early 20th century. So what about since then? Well, in this period of 1918 to 1947 is what is sometimes called the neo-Darwinian synthesis. So this has taken Darwin's theory and was merging with Mendel's laws of inheritance and the mathematics of population genetics, and kind of that's shall we call the theory of evolution that's pretty much taught today. So that's the neo-Darwinian synthesis. Well, the middle of that time is when the electron microscope was invented. A little bit after that was the DNA double helix was discovered, and then the DNA code was broken. And this showed the information basis for life. Of course, you can ask the question, where did the information come from? But it was in 2001 that the human genome, that is the genetic code for human beings, was decoded. And since then, there's been an explosion of diagnostic and analytical tools. One of them is called cryo-electron spectroscopy, where they can actually freeze molecules and actions, and they can actually elucidate the structure of proteins. So with all this explosion of diagnostic tools, as we're able to investigate things that are smaller and smaller, what have we found? Well, Michael Behe, a biochemist, said, "So what have our powerful new tools showed us about the physical basis for life? The details of studies fill libraries, but the overarching picture is one of fathomless elegance, a seemingly never-ending parade of sophisticated structures and brilliant organizational arrangements and well-nigh incomprehensibly complex systems." So you can see a diatom in the left using electron microscope. So 5 micrometers is on the order of a tenth of a thousandth of an inch, and you can see the features on that. To the right are the structures of protein complexes laid out. So each color is a different protein, and the proteins fit together like pieces in a puzzle to perform functions and so forth. And you might say they're the parts and the machines of life. Here's an example of a molecular machine made of proteins, and this is the flagellum of the bacterium. So the flagellum has a propeller to propel through liquids, water, and blood, and all sorts of other things. And these propellers are powered by little motors. And so here's a picture of what a flagellum looks like. And you can see the electron micrograph of one. So it includes the elements of a machine. You have a rotor, which is the part that's attached to the shaft. And then you have the stator, the stationary part. And it's the force that exerted by the stator onto the rotor that causes the whole system to rotate. And then you can see shafts, you can see bushings, you can see a universal joint. So this allows the rotation axis to turn around a corner so that the flagellum can whip around a different axis. So you look at this and you wonder, you know what, this seems to have all the hallmarks of design. And so the story behind Michael Behe himself is rather interesting. So Michael Behe, he's a Roman Catholic, and he has seen no conflict between Christianity and Darwinism. However, his interest was piqued when he studied the bacterial flagellum and other biological structures and realized that they could not come about through small incremental steps resulting from random mutation and natural selection, which is the foundation of Darwinism. And in fact, it's probably the only way that life could occur is through these small random changes through time. And if you have big step changes, it's just kind of akin to miracles, and so that's a bit of a problem. So he realized that these structures cannot be built up from small pieces with incremental changes, because they were basically all or nothing entities. And so he came up with this idea called irreducible complexity, that there are structures, assemblies, that you can't make them any simpler and have them still work. And so the question is, how do you get those structures to begin with? They may not be perfect structures. They might not be the ideal designs. You have to ask the designer for that, what their intent is. But they can't be built up by just random assemblies, piece by piece, until all of a sudden start working. Because if it doesn't work from the beginning, it's not going to continue to progress. So that's the whole idea of irreducible complexity. Behe gives a lot of examples of these kind of structures. We see the flagellum here. Now this little flagellum, it's rotating machinery, as some of you are involved with. For E. coli bacteria, that little propeller spins at 18,000 RPM and pushes the cell at 15 times its body length in a second. And the speed records that are set for other bacteria, these things are spinning at 100,000 RPM. And those things would be moving, of course, much faster. So amazing little nano machine, and I'm sure human engineers wish they could develop something like this. So there's more wonders with the flagellum. For example, on some bacteria, there's an idling clutch, disconnecting the flagellum from the motor itself so it can stop. There's also an emergency release system. So if the bacteria is facing starvation, it can minimize its energy consumption by disabling and ejecting the flagella. So those of us who are engineers wonder how on earth, or I don't know about you guys, but I marvel at these kind of things. So design is not necessarily a simple thing. Here we have a rocket. A rocket consists of a bunch of systems. And the systems consist of subsystems and subsystems of parts and components and stuff like that. So how do you actually go about building a rocket? Well, first of all, you need design drawings, but that's just an idea. That's what that is. But then you need materials, and you need fixtures and tooling, and you need fabrication. You have a fab shop at work, several of them. You have to have mechanics that perform assembly and checkout. And then you have quality control, the folks looking over your shoulder to make sure that mistakes are found and corrected. And then you have repair and maintenance, the people coming around and fixing things when they get damaged. And then you have disposal. You have the garbage trucks that come up and dispose of things when they're no longer needed or have reached the end of their useful life. So it's not just a matter of what, which is what be the design of the protein or the cell itself. It's not just the what, but it's the when, where, and how. And of course, all this is done by the cell autonomously. This thing is so small you cannot see. It does all this automatically. So I'll introduce you to this thing called functional coherence, and that's a hallmark of design. It's where all the functions that you have functions that support other functions, that support bigger functions. And all these little pieces, parts, have to work together to form the overall system so that it can perform its function. So here's an example of a tractor motor from a parts catalog, and you can see a gear. Well, the gear itself, a single part, has functional coherence. It has a pilot for centering it on the shaft. It has a flange for connecting it to the end of the shaft. Then it has bolt holes for bolts to attach it. And then it has an inner rim and spokes and an outer rim and teeth. And all those pieces are not random. They all have to fit together in a coherent way. And so that's functional coherence. And so now you put that on the shaft and it has to coherently work with all the other parts. So engineers recognize this right away. They see this, they see design. What about at the level of life? So life shows functional coherence at the deepest level. So let's look at protein. Proteins are the machinery of life. They're made up of chains of amino acids. Amino acids are something that, of course, we get in food, but they're hooked together like Lego bricks to form long chains. And as these chains, if you have the content and the order of the amino acids right, then the chain automatically folds up into a functional protein. So here's an example. So the protein is once it's completed, it folds up. You might say the part kind of self-deploys, and it's that shape that puts the right features at the surface that enables it to perform its function. So here's an example of a 3D ribbon diagram of a protein. This is myoglobin, which stores oxygen in your muscles. And you can see the protein there. A little red dot is a diatomic oxygen molecule. So the protein has to grab onto that, hold it. It also has to release it at the right time. So without functional coherence at the deepest level in the order of those amino acids, you won't have a functional protein. So here's a picture of another functional protein. This is called kinesin. It's a little motor. It's used for dragging larger things like protein complexes, organelles, and things like that along cables, which are other protein structures from the nucleus to the outer part of the cell. It's a little train, and then there's another motor protein that carries it in the opposite direction. So these are pretty amazing. So the chance of forming a functional protein from a random selection of amino acids, it has been experimentally determined to be 1 in 10 to the 74th power. So that's an extremely small number. So it's not like you can just throw a bunch of amino acids together, shake them, and out come proteins. No, it requires information. You have to have information on what amino acids to select and how to hook them together in order to form functional proteins. So information must be stored, it must be retrieved in order to be able to make the functional proteins of life. Well, where's that information? That's where DNA comes in. DNA is a ladder-like molecule. It twists into a spiral staircase structure. So each step or each rung on the ladder represents one of 4 letters. And 3 consecutive letters or rungs form a codon, and a codon indicates one of 20 amino acids. And so a gene is a string of codons that specifies an amino acid chain that will fold into a functional protein. That is where the information is stored for forming proteins. So DNA is an information-bearing medium on which it is stored. So information is independent from the object on which it's stored. It can be copied. It can be transferred to another medium. For example, in a cell, it's transferred to DNA and then to proteins. Or it could be on a hard drive, or it could be on a piece of paper, or a book, shall we say. And information can be degraded or destroyed, but the question is, can it be created through blind processes? Can it really be created apart from design and intelligence and so forth? So the question is, now we've talked about having the plans is one thing, but implementing the plans is something else. So how does the cell take this information and turn it into a protein? So let me stop presenting and show you something else.
What you are about to see is DNA's most extraordinary secret: how a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body. When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
that's how DNA is turned into flesh and blood, as they say. So what about the information in the DNA? It really has functional coherence. It's language. So it includes an alphabet, includes words, it includes structure and meaning. So you compare that to English. So suppose you have a half a page of random keystrokes. What would be the chance of that consisting entirely of English words? So of course, there'd be much more information than this in a single molecule of DNA. Well, the chance of a half a page of random keystrokes containing English word is 1 in 10 to the 599th power. So to put that in perspective, the number of possible events in the entire universe has been estimated. You can find in my notes. I didn't estimate it, but I got it from someplace else. It's 10 to the 120th. Okay, so how many universes does it take to for you to randomly have keystrokes make a half a page of English words? It's 10 to the 479th universes that would be required, an unimaginably large number. Well, they're all unimaginably large. I wanted to show this video. One of the most amazing thing that cells do, or life does, is it reproduces. So you engineers, I'd like to know how many machines you've designed that can make themselves. Well, I can pretty much guarantee you'll say none. But that's what cells do all the time. They divide in half, but somehow the information doesn't divide in half. After they divide, then they grow, and then they divide again, then they grow, they reproduce, they copy themselves. It's only done on individual cells, but for creatures, multicellular creatures. So here's a mouse, and this is a mouse embryo, different views of the embryo as it grows, and you can see how the cells reproduce and how they move to basically form a mouse. So those all started from the same cell. As the cell divided and grew, and then they started taking on separate functions and moving to other parts of the cell to form the various organs and so forth. So to me, this is just absolutely incredible. The amount of engineering, the amount of intelligence that is behind life is just absolutely unimaginable. So I think I'm going to move on because we're just about out of time. So what's the conclusion? I just want to point out, what's that have to do with Scripture? Well, the Bible says that we are fearfully and wonderfully made, and the psalmist said, probably 3,000 years ago in Psalm 139, he said, he's talking to God. He says, "For you created my inmost being; you knit me together in my mother's womb. I praise you because I am fearfully and wonderfully made. Your works are wonderful, and I know that full well. My frame was not hidden from you when I was made in the secret place, when I was woven together in the depths of the earth. You saw my unformed body, and all the days ordained for me were written in your book before one of them came to be." So you are designed. That means you have a designer. The designer is God, and you have purpose. And where do you find that purpose? You find it from your designer. So I want to share one other passage with you. So where does life come from? Well, we NASA like to look for life elsewhere, and we look for the ingredients for life, the things that are necessary for life. So we look for carbon, we look for oxygen, we look for different elements. We look for water. Follow the water, we often say, as we go to other planets and find oceans underneath ice crusts on moons around in the outer solar system. So always look at the ingredients necessary for life. But one thing that we never mention is that one thing necessary for life is previous life. We don't know a single counter-example. You came from your parents came from their parents. All life we have today came from previous life. So where does life ultimately come from? It comes from God. He's the one who created life because He is the designer. And so I want to end with this passage in John. So this is the Lord Jesus talking. He says, "Very truly I tell you, whoever hears my word and believes him who sent me has eternal life and will not be judged, but has crossed over from death to life. Very truly I tell you, a time is coming and has now come when the dead will hear the voice of the Son of God, and those who hear will live." Why? "For as the Father has life in himself, so he has granted the Son also to have life in himself." So the Father has life in himself. He didn't get it from any place. He's eternal. light. He is life in himself. He has life in himself. And so that's where life comes from. And he has granted the Son also to have life in himself. And he has given him authority to judge because he is the Son of Man. Do not be amazed at this, for a time is coming when all who are in their graves will hear his voice and come out. Those who have done what is good will rise to life, and those who have done what is evil will rise to be condemned. And I say, if you want life, you go to the source of life, which is God. And that means going to his Son, the Lord Jesus Christ. All right, so we're going to continue on with this next week. Class isn't over yet, by the way. Don't go running to the doors just yet. So, but we're going to see more amazing examples of design, and a lot of it's in the area of reproduction, how cells reproduce and so forth. And then we're going to get into one of the things I want to discuss, or what are some of the objections that are raised against design. So I have spent some time on that. I want to talk about this concept of the breath of life, so tracing this through the Old Testament. So in Genesis 2, it's first mentioned how, God formed man from the dust of the earth, and he was just a lump of clay, I like to say. And then God breathed into his nostrils the breath of life, and the man became a living being. So this phrase, the breath of life, is really an idiomatic phrase. So God gave him life. He's the one that made him come to life. This isn't just reserved for humans. This is reserved for animal life too, and I would say probably all life, organic or plant and fungi and bacteria and all that kind of stuff. He says, when it comes to the flood, God says, I am bringing the flood water upon the earth to destroy all flesh, in which is the breath of life from under heavens. Everything that is on the earth will perish. The next one, he talks about 2 by 2s, all flesh, going on into the ark, and they're referred to as the ones that have the breath of life. So the breath of life is that life that God gives. It's his breath, in a sense, I think, is the biblical image. And you can see in Genesis 7, well, all those that had the breath of life in their nostrils died, okay? And then in Job, he talks about that the breath of God is in his nostrils. So it's like the breath of God is similar to the breath it's synonymous with the breath of life. And sometimes they would say that God who gives me breath, right? So God is the one who's giving you life right now. He's the one who's giving you breath. And you can see that again in Job 33. The Spirit of God has made me, and the breath of the Almighty gives me life. But our life is a derivative life. So you see that in Isaiah. It says, stop regarding man, whose breath of life is in his nostrils, for why should he be esteemed? It's in our nostrils. In other words, you can just close that, in a few minutes you're going to keel over, right? So but God has, don't try that experiment, okay? But anyway, God has life in himself. We don't. God, he gives life to us. He gives us the breath of life. And in Revelation, it talks about, remember the 2 witnesses. So even this biblical allusion is used in Revelation 11, where you have the 2 witnesses that are killed because they didn't like, the people of the world didn't like what they were saying. And so it said after 3.5 days, the breath of life from God came into them, and they stood on their feet, and great fear fell on all those who were watching them. So God, life comes from God. It's his breath in our lungs, so to speak. So just remember that. And, the bottom line is that and this is what I don't want to lose focus on, is that life is designed. You are designed, and it's obvious if you look at it that it's designed. And we'll get into more of that evidence next week. And design means designer, and designer ultimately means God, and it also means purpose. So with that, our time is up, and we'll continue this discussion next week.